Hi there, everyone. We are so happy to have you, whether you are joining us here today or will be watching later on our Cure GABA-A YouTube channel. We ask that you please mute your mics if you have not done so already, and include any questions that you might have in the chat box below. We'll have time to answer questions at the end of the presentation. My name is Monica Joanna Elnakabe, and I'm the founder and CEO of Cure GABA-A, a nonprofit organization with the sole focus to develop potential therapies for GABA-A variants by partnering with world-renowned researchers in GABA-A and the Organization of Patients for Clinical Trial. Today, we welcome Dr. Kirill Zavalin, a postdoctoral fellow at Katty Kang's Lab at Vanderbilt University. Dr. Zavalin specializes in genetic epilepsy and brain development with a particular focus on GABAergic neurotransmission. His expertise lies in unraveling the complexities of epilepsy, aiming to contribute to the advancement of biomedical neuroscience research. I'm handing it off to you, Dr. Zavalin. Please share with us the role of GABA during development.
Thank you for that wonderful introduction, Monica. Okay, so like Monica said, my name is Kirill. I am a postdoctoral fellow in Dr. Kang's lab, and we're at Vanderbilt University Medical Center. But a good amount of the work that I'll present today was done during my thesis research, which was also at Vanderbilt, but it was done in the laboratory of Dr. Andre Lagrange. So a good amount of it will be perhaps more basic research about the role of GABA in development. And so the relevance of it for specific disorders associated with specific mutations in GABA receptors and GABA receptor subunits, we'll have to kind of infer from that. But I think it'll be really interesting for this audience. So first, I want to just briefly set the stage and explain kind of what the cortex is like and what development is like. So today, I will focus specifically on development of the mammalian cortex. So probably most of you know what that is, but it's basically the part of the brain that's responsible for the higher order functions. And you can see it here labeled anatomically. And we don't have to get too much into the structure of cortex. But I just wanted to touch on the fact that it has two main types of neurons in it. One type are these principal neurons, which are the majority of them. You can see them here in the image on the right in black. And they are excitatory. So whenever they contact another neuron, they make it in turn fire. So they just basically talk to each other, and the signal keeps propagating as they're talking to each other. And they can send these signals over long distances. And then a minority of neurons in the brain are GABAergic interneurons. They release GABA. That's the way they communicate. There are many subtypes of them. And they're called interneurons because they only communicate with local neurons, and they regulate their firing by inhibiting them, by using GABA to inhibit them. And this is a very important population of neurons. So when these neurons aren't working correctly, that's been found in a number of diseases like epilepsy, but also schizophrenia, autism spectrum disorders, and other disorders of the central nervous system. So when cortex develops, I won't go through the entirety of the sequence that occurs, but some of the general events that occur is one, that the neurons that will populate the cortex, they have to get there. So at first, they will migrate. And for these two different populations of neurons, that happens differently. So see if I can get a laser pointer here going. So if this is here, cortex, so the glutamatergic excitatory neurons, those projection neurons, they're born by the ventricle, basally here at the bottom, and then they migrate out to form the cortex. And here they're shown again in red right here. They're migrating up and out, and they form the cortex in this cortical plate labeled here as CP. And then the interneurons, they're born kind of lower down by the striatum, and then they perform this elaborate migration where they migrate out and then in these two migratory streams, they'll migrate, and then eventually they will take a turn and go into cortex, and then they'll settle cortex. And so in the end, eventually you'll start forming cortex, deeper layers first, you can see here on the right, and then superficial layers last. So first you form like layer 6, then 5, then 4, then 2, 3, and then the end, you end up with 6 total layers. These populations of cells will be mixed at the end, but they follow different migratory journeys. And so after these cells migrate, then there are several other very important processes for them to form those circuits of neurons that will be working in the mature brain that you need. And those two processes basically are extending neurites. So neurites are little projections that neurons have, and that they contact other neurons of these projections and they communicate to one another. So they will extend these neurites, and then they will form synapses. And of course, synapses are the sites where one neuron will release neurotransmitter like GABA or glutamate onto another neuron. So that's the point of contact, point of communication between two neurons. So as development occurs, and in this case here on the left, this is actually in a human for the stages that are written down there. But you can see over the course of development, the projections that these cells have, the neurons that they have, become more and more elaborate, especially at first. Then they actually come down a little bit. So there's at first you get more and more elaborate, and then there's a period of refinement. And similarly for synapses, which is shown here in a simple schematic on the right, where at first you have an axon, so that's a way that one neuron will communicate with another, and it will actually form a synapse on a dendrite. So this is one neuron here, an axon, and this is a dendrite of another neuron. So this neuron would be releasing a neurotransmitter, this neuron would be accepting it, and then this would be a synaptic contact. This would be a synapse. And so you can see here, a lot of synapses are formed at this middle stage, and then some of them are pruned away and refined. And that's a general process of development. And of course, a lot of this we've studied in rodent models, and we used it to predict how and to understand how it occurs in humans. Those timelines are different, and that's what I'm showing you here. Basically, on the left is a summary of what I've shown, and you have both a mouse version and a human version. So for instance, the human cortex forms in a similar fashion, but it's got a few other steps that make it slightly more elaborate because it's a more dense cortex. Obviously, our executive function, higher order function, is higher than that of rodents. We have a lot more cortex to accommodate for that. And so then on the right also is the approximate timeline of how these processes occur. So you can see that here, for instance, neurogenesis. So that's going to be the birth of neurons. That's going to occur before birth, pretty much for both mice and for humans. I mean, it looks like a little bit of neurogenesis is marked here as well in mice after birth, but most of it's going to happen before birth. But then some of these other processes, such as synapse formation, in humans that can last for a very long time. And then in mice, that pretty much only lasts 3 weeks after they are born. And some of these other processes as well. And you can see also it's spread out pretty broadly because, for instance, in humans, sometimes it's not known with perfect precision. But so, yeah, and then after these are formed here in green, then you'll have a time of synaptic pruning. That's when you have that refinement, or some of the synapses start going away. And so that can go for an even longer period of time. And I'm going to talk about development and developmental processes in mice today. So this kind of general timeline can be used to approximate how that would translate to humans. Although exactly how to translate animal studies to humans, that's an ongoing question, because it's different for many different programs and aspects that you will look at. So now that we've generally reviewed developments, I wanted to go back to GABA. So GABA, actually, you probably all know it as a neurotransmitter that inhibits other neurons in the adult brain, very important for that role. But it also actually is important for development. So, for instance, out of some of these general processes that I've described, GABA has been shown to be involved in dendrite extension. So dendrite, that's a type of a neurite. That's basically a neurite that is used by a neuron to receive input from other neurons. So it's been shown to help extend those dendrites during development. And here it's shown also, you can see quite dramatically when GABA is not able to give that signal, these neurites that normally would go up at the top of cortex, they are stunted. GABA has also been shown to aid in synapse formation. For instance, in this experiment, GABA has been released by uncaging. So it's a really cool technique where they basically have GABA in a little parcel and they shine a laser and they suddenly bust it open and all this GABA is released. And when they did that, they noticed that they were able to form new synapses on like neurons where they did that. In this other study, it's shown that early on during development in cortex, a whole set of neurons will fire together because they all release GABA to one another, seemingly, or at least GABA is released onto them. So they will fire together like this in concert. And this actually helps form some of the early circuits because by firing together, it kind of trains those neurons to work together. And in this other experiment, again, it showed that synapse formation was dependent on GABAergic signal. When they got rid of that signal, as we basically see here with black, when they got rid of that signal, the number of synapses went down in this particular experiment that they did. And it's also, GABA is also important for migration. In particular, has been shown to be important for migration of the interneurons, of the GABAergic neurons. So as I showed you earlier, interneurons, they do this very elaborate migration where they start out, they're born here by the ventricle, and then they migrate out and throughout cortex, and then they populate it. So in these experiments, and this is an experiment where they took basically a slice of the brain from a mouse, and then they cultured it, and they looked at how these cells migrate there. They noticed that GABA is very important. For instance, if they block it with a GABA-A receptor antagonist by bicuculin here, then these neurons don't go there. And then on the other hand, if they give a drug that potentiates GABAergic signals, so a positive allosteric modulator diazepam, then more neurons migrate up there. And a couple of other experiments also basically showed that for migration of neurons, of GABAergic neurons, as they're migrating here in the cortex, this GABAergic signal was very important. And that's what these two experiments show. Basically, they can't migrate there. And so how does this actually occur? And I'm going to talk about two aspects that we know of, at least, that allow GABA to be an important cue during development. So this is what a typical GABA-A receptor looks like. So it's made up of 5 subunits here. And we'll talk about that some more later. And it is basically an ion channel, in that whenever GABA, neurotransmitter GABA binds, and here you can see GABA binding, these big pink balls are GABA. When GABA binds, this channel will open. And then the ion chloride primarily will move through that channel. And that's how the signal is passed by that receptor. So typically in the adulthood, what happens is because there's so much chloride outside of the neuron and pretty little inside, when you open that channel, chloride rushes in. And so that's negative charge, because chloride is a negatively charged ion, negative charge moving in. So that ends up inhibiting the neuron. And that's what basically inhibits the signals there. That's what prevents that neuron from firing. That's how the inhibitory neurotransmission works. But the case is different in development. Basically, you can have a polar opposite effect simply because the concentration of chloride is different. So now you have more inside the neuron. And so then when you open this receptor, the chloride will actually flow out. And then you can have an excitatory effect instead. It's almost like, flipping a switch in different directions. It's kind of crazy. And the reason this occurs, there are thought to be multiple reasons, but the biggest one is thought to be because there's this particular transporter called potassium chloride transporter 2. It's KCC2 is what its what its abbreviation is and what it's called. But that's simply because it transports 1 potassium and 2 chloride. But so what it does is it's able to take chloride inside of a neuron and pump it out. And it is expressed quite highly in mature neurons, but quite low levels of it are expressed during development. So because it's not working very hard during development, because there's not much of it, and there are other regulations that keep it from being particularly active during development, it doesn't extrude any chloride, so the chloride is able to accumulate in these neurons. There's also another transporter called NKCC1 that actually helps move chloride into the neuron as well. So as you can see here on the bottom, during development, there's a lot of chloride here, shown here, as if chloride is kind of water filling up this bubble. And there have even been experiments, including here at Vanderbilt, that basically show that if you get rid of this transporter, that chloride will stay high in those neurons, basically. And then GABAergic responses continue to be excitatory. So KCC2 is very important. Here, I'll actually jump to this slide. So KCC2 is very important for having GABA be excitatory, because basically, when there's no KCC2, there's only NKCC1. There's a lot of chloride in the neuron. So when you open the GABA-A receptor, then you'll have an excitatory response. And what that ends up doing is it activates these voltage-gated calcium channels. And this calcium that goes in the neuron, it ends up promoting a whole number of different events. It can help change the skeleton of a neuron, to have it send out neurites, to help it move during its migration, to help it form a synapse, stuff like that. And this is a pretty technical detail, so maybe not as important to remember this, but I figure I'd still provide it if you're curious as to how it actually works. And then as the neurons start maturing, they'll start expressing KCC2, which will extrude the chloride, will pump it out. So then when you activate these receptors, the response would be the opposite. Chloride will go the other way. So actually, it will inhibit the neuron from being active and signaling to other neurons. And it could actually also oppose these developmental processes by preventing activation of those calcium channels and preventing influx of calcium that could help with the developmental processes. So the response is very different. And now to go back to this other slide. So one of the things I've done in my thesis research is I've looked at how during development KCC2 is expressed. And so this is in mouse brain sections, going from, so E would stand for embryonic and P would stand for postnatal. So going from about the middle of embryonic development up until the brain is almost in its mature form at postnatal day 19. And you can see that at first there's in the cortex very low amounts of KCC2. Levels start picking up very slowly, very small amounts, and only by the end of the second postnatal week in rodents, pretty much when synapse formation is like starting to really kick in, and some of those initial developmental processes are kind of coming to an end. Do you actually have significant levels of KCC2 being expressed? To go back to the slide that I've showed you earlier about how GABA promotes neuronal development. Well, in these experiments, it's also been specifically shown that the excitatory aspect of GABA was important for it promoting development. So for instance, in this experiment where they looked at how GABA was important for dendrite extension, well, they had that dendrite extension be stunted when they knocked down one of the transporters that allowed GABA to be excitatory. Okay, and similar here, basically they overexpressed KCC2. So we just talked about how KCC2 usually comes online during the later stages of development. So when they had it come in too early, that seemed to stunt the formation of these neurites here. In this case, in this experiment, I don't have it singled out because they didn't actually prevent GABA from being excitatory, but they actually confirmed that GABA was excitatory and actually allowed that calcium influx to occur. And that's in blue, this trace you see, that's what they're seeing. They're seeing basically every time these cells activated, they had this influx of calcium. And if they gave a blocker that blocked GABAergic signaling, all of this basically went away. And in this experiment as well, when they prevented a transporter from allowing chloride to be high in the neurons, that's when they saw a decrease in the synapse density in these neurons that they studied. And the other cool thing about it is you may have noticed when I was showing you expression of KCC2 that while it is quite low early during development, so here, for instance, at postnatal day 0, so right as these mouse pups are being born, it looks like there are individual cells here that do express it. And it turns out that these cells are the GABAergic interneurons. So not all of them, but some number of them seem to express KCC2 early on. And basically, we could confirm this by using a marker for interneurons. So it's called the DLX5 marker. That's a promoter that they use. And basically, if you look, then this early expression seems to be restricted to these specific cells. Later on, as the cortex develops, that's not really the case. You can see there's a lot of KCC2, and it's present in these green interneurons, and it's also present in the space surrounding them, and it's not specific at that point. But earlier on, it is quite specific. And here's another image to kind of drive that message home. So this is a confocal image, so really zoomed in, nice resolution. So you can see KCC2 here being expressed at the membrane of these two neurons, which are also expressing this green marker that marks them as GABAergic interneurons. And so GABA, and specifically the excitatory aspect of GABA, also seems to be very important for development of these specific cells. For instance, going back to this slide on the migration of these cells, well, this study didn't look at it, but in this study in the middle, when they just either gave a drug that prevented GABA basically from being excitatory, it affected one of those transporters of chloride, then they really reduced the migration of these neurons. Or if they prevented those calcium transients from occurring, they also really tanked the migration rate of these neurons. And in this experiment, they simply overexpressed KCC2. So basically, they made the cells make KCC2 before they would normally make KCC2. And when they did that, these neurons would no longer migrate into the cortex the way they normally did. So the expression of KCC2 that comes online at the appropriate time, not too early, is very important for these cells. That GABAergic depolarization, that excitation provided by GABA, is an important signal during development, specifically for migration. Could be for other aspects as well. They just haven't been studied. So actually, one of the projects that I undertook as a graduate student is I wanted to know what role KCC2 plays in these cells. So I created a mouse where we deleted KCC2 from interneurons. So we know that if it comes too early, it's really bad for these cells. And we decided to see what happens if it doesn't come at all, if maybe it has to happen just at the right time to help transition from this kind of developmental GABAergic signal that's excitatory to a mature GABAergic signal that's inhibitory. And what I found out is that these mice, they were quite sick. Again, they're missing KCC2 only in the GABAergic cells. And they were smaller in weight than their siblings. So wild type here would be a normal mouse, and CKO here would be a mouse that's lacking KCC2 in the GABAergic neurons. And you can see they're smaller in weight. They're shorter in length. and they would die much earlier. So here's a survival plot. So see on the x-axis, you have the days as they go, and you have percent survival on the y-axis. And you can see that most of the mice that don't have KCC2 in their interneurons, they would die between the third and the fourth postnatal week, somewhere in there. So they really wouldn't typically live a month out. And the way that they died would be because they had seizures. So I actually did monitoring with a video camera at night to see that. I don't have that here, but what I do have here is also to show that they had susceptibility to seizures. So if you were inducing a seizure, then these knockout mice, they would get a seizure much faster than their litter mates that were normal, basically. And the other thing I noticed is if you look at how the GABAergic neurons distribute throughout cortex. So again, we talked about how KCC2 seems to, well, GABAergic signals seem to be important for regulating migration of these cells. When KCC2 is missing in these neurons, the distribution in the cortex and the end seem to be altered. So I had more studies that I'm just showing here, but just looking at all interneurons, for instance, you can see there are more cells in layer 5 in this knockout that's missing KCC2 in all GABAergic neurons. I do have to add, though, that I kind of thought the effect would be even stronger, and it wasn't. And so regarding this particular process, there's still a lot to be studied. But I do think it's clear that KCC2 is very important for this particular population of cells. Okay. So we talked about KCC2 and the fact that GABA is excitatory, and that's a very important aspect of GABA in development. But it's not the only. And so how else does the GABAergic system adapt to its different roles in a developing and mature brain? So let's go back to this image of a GABA-A receptor. So it's made up of 5 different subunits. So it's a heteropentamer, hetero for different, pentamer for 5. These are typically 2 alpha, 2 beta, and then a gamma or a delta subunit. And then the composition of these receptors, the subunit composition, it can affect a great deal of things. It can affect where in the neuron these receptors localize, where they go. It can also affect a lot of the properties of these receptors, how they might signal. Just a bit of foreshadowing, but changing that composition is how GABAergic signaling could evolve from mediating a developmental signal then to later mediating signal at a mature adult inhibitory synapse. So one thing I mentioned is that the combination of these subunits, it can affect where in a cell the receptors localize. So there are 12 commonly occurring subunits of GABA-A receptors. That's alpha 1 to 5, beta 1 to 3, gamma 1 to 3, and delta. And there are some others out there. There are 19 known total, but these are the main ones. And I'm also not mentioning alpha 6 here because that's mainly expressed in the cerebellum, and I'm talking about the cortex here. So out of the synaptic receptors, they typically contain a gamma subunit, often specifically gamma 2, and then they'll contain alpha 1 through 3, alpha 1, alpha 2, or alpha 3, and then some beta. Often it's beta 2 or 3. And then the extrasynaptic GABA-A receptors, they're going to either be combinations with the delta subunit, because this is because the gamma 2 subunit seems to be important for actually tethering those receptors to the synapse. So if instead of gamma 2 you have a delta, it often can be extrasynaptic. And that's often partnering with alpha 4 subunit. And then there are these subunit combinations that have alpha 5, beta, and gamma 2. And even though they have gamma 2, these guys can be either synaptic or they can also be extrasynaptic. Often they actually are extrasynaptic. Other aspects that are determined by the composition of these receptors is the sensitivity to GABA. So here, for instance, you see on this plot, you have the concentration of GABA on the x-axis. So that's how much GABA you're adding onto a cell that's expressing these receptors. And on the y-axis, you have the currents that you can record from activating these receptors in the cell. So you can see that, for instance, for a combination like alpha-4, beta-2, delta, you need a very low concentration of GABA to activate them. And these are these extrasynaptic receptors. So they activate to a very low concentration of GABA because they're not sitting at the synapse, they're sitting far from the synapse, and they activate with just a low concentration. On the other hand, something like an alpha-1, beta-2, gamma-2, so that's the kind of combination you normally see in a synapse in the adult brain. Here, the black line. It requires actually more GABA, but that's normal because it's sitting at a synapse. So whenever a neuron sends a signal, it sends suddenly a bunch of GABA right there, right at the synapse, so it can activate. And then you can see some other combinations, though, like this alpha-3, beta-3, gamma-2 combination. It requires even more GABA to activate. And there are other properties, other ways in which these different combinations change how a signal passed by a GABA-A receptor will be different. So in these traces, for instance, basically, again, the combination of subunits is being expressed in a cell that's grown in a dish. So in this very isolated environment, we're able to record what a trace, a GABA-A receptor mediated current would look like. And basically for all of these, during this period of activation right here, we're adding a little puff of GABA like this. And basically what you see is they all activate to different degrees. So the kind of strength of signal that they can pass is different based on the composition. And then how quickly they desensitize. So how quickly, even though there is GABA, the current here diminishes, the strength of the signal diminishes. Also, it differs. So for an alpha-3 containing receptor, for instance, it's clearly not diminishing as much. It's not desensitizing as much as far as alpha-1. And similar could be said about alpha-5. So basically, if you keep giving these neurons GABA, they will keep being able to react to them if there's not a lot of desensitization. Versus in this case, there's so much desensitization, if you keep giving it, they won't be able to. And there have been a couple of big studies that have shown this, including this one here by Picton and Fisher. I really won't go into the detail here, but basically they express different combinations containing different alpha subunits. And they showed that the currents mediated by these combinations are different. So this is one of the ways that GABAergic signals can be modulated and changed just a little bit to form the different needs that might occur, either because it's development versus later in life, or maybe even a specific function or specific localization. Like, for instance, some of those subunit combinations that are expressed later in life, like alpha-1 receptors versus alpha-2 containing receptors, they can be expressed in different types of synapses because they do slightly different jobs, slightly different functions. And so one of the things I did as a graduate student, and I did this with other researchers, is I looked to see how the composition of these receptors changes as development progresses. And so that's what I want to show you right now. So first thing we did to investigate this is we looked at the major subunits, at least the ones that we had good means to identify and detect. And we saw, as development progresses, how the amount of these subunits changes in the cortex of mice. And so for each of these plots, on the x-axis, we're just going through these different developmental time points, kind of similar to how those overlays that I showed you, basically at first starting from an embryonic age, 14 days as an embryo, and then to postnatal age P25, so with birth being right around here, right before P1. And basically, when we did this, we kind of sorted what we observed into two groups. Subunits that are expressed early, and then subunits that are expressed later. And so the ones that we saw that were expressed early were particularly alpha-3 and alpha-5. And we also saw beta 3 and gamma 2. And by the way, when I say expressed early, it doesn't mean that they weren't expressed later. So some of them, like alpha 3 and alpha 5, indeed, they peaked during development, and then they kind of came down to a lower plateau, but that was still a prominent one in the brain that was almost an adult brain. But for some of these other subunits, particularly gamma 2, it just kind of took off and skyrocketed, went even higher in the adult brain. But still, that was in contrast to these other subunits that are expressed late, that was sorted as late. So these were actually quite low during embryonic and early postnatal development, and only started coming online as the brain became maturing and transitioning to truly being like an adult, mature brain. These are the Western blot experiments that we've done. So basically, we just took all of the tissue from the cortex and we homogenized it, and then we just looked how much protein there's in there. But then we also looked, in the way that I showed you KCC2 expression, we looked how the signal changes if you look at pieces of tissue. So here you have pieces of cortex from a mouse, progressively again from embryonic time points up into late postnatal development. And this is on a heat map scale. So basically, the brighter the colors are, you can see here the bar for it, but basically the brighter the colors are, the more KCC2 there is there. And you can see for gamma 2, early on in embryonic development, there's a lot of it. I mean, there's a lot of it also in the adult brain, and that's of course because gamma 2 is a subunit that's present in so many GABA-A receptors that are at synapses. So both late and then early, even when synapses aren't formed yet, there are already receptors of gamma 2 in them. And likely the subunits that are partnering with this gamma 2 subunit at these early stages are alpha 3 and alpha 5, because they also come online very early, starting from embryonic development. Both of these subunits are present, and they seem to be quite important for the developmental processes that are promoted by GABA. Now, a lot of it still remains to be investigated. Like, for instance, all those studies that I showed you, which still, there's a good number, but there could be a lot more. For most of those studies, people aren't specifically looking at what subunit is mediating, but they just show basically that GABA is important. But there have been some subunits that looked some studies that look specifically at what GABA receptor subunits are important for development. So for instance, there's this one study by Brady and Jacob in 2015, and they saw that specifically alpha 5 containing GABA-A receptors, alpha 1 containing GABA-A receptors, they promote dendrite and synapse formation. They had this cultured neuron setup where they were looking at how the contacts between those neurons are forming. Yeah, so alpha 5 containing GABA-A receptors seem to be particularly important there. And then in another work done by my thesis advisor, they looked at how alpha 3 containing GABA-A receptors can be particularly important during development, because they're able to sum up repetitive stimuli. So for instance, in a case of maybe a synapse being developed, a neuron can detect that a particular signal keeps coming from one source because it's summing up repetitive stimuli. Or in the case of a neuron migrating or extending a neurite, you can detect that there's presence of GABA coming out from a particular source, and that can be guided to go in that direction. And that's, again, when we talked about the different properties where different receptors with different combinations can have, the fact that you really need a whole ton of GABA to activate these receptors, specifically the alpha 3 containing ones, and the fact that they don't desensitize, meaning like if you keep giving them a lot of GABA, they're not going to be strongly losing the response they get to GABA. That actually predisposes them to be able to act in this manner, and a different combination would not be able to act in this way. Now, this is still all conjecture, but basically in the scientific community, these are some of the hypotheses that have been made to explain why these particular combinations might be important for development and how they might be able to drive some of these developmental processes. But a lot of this still needs to be investigated. So that's alpha-3 and alpha-5. But I'll go ahead and move on to some of the other subunits. So for instance, alpha-1 and alpha-2. These seem to be very important now for the adult brain. You can see there's not a lot of them at early postnatal and embryonic time points, but they really increase in expression as the brain starts to mature. There's actually an interesting pattern of expression we saw with alpha-2. You can probably see, or just in like layer 1 and the top layers of cortex, even at early developmental time points, we start seeing it. And we don't really know what the significance there is. Also, with a lot of these subunits, we've seen some patterns like certain parts of cortex, like layer 4 in particular, seem to start expressing these subunits early, and then the rest of layers start expressing it later. And the full significance of that is not entirely known, except that these particular parts here are the first to start, they basically start forming a connection with thalamus. So thalamus is an underlying subcortical structure that relays a lot of, for instance, sensory information, like your sight or touch information. It will relay it up to the cortex, whereas now the cortex is going to be the high order structure. And this layer 4 is where that contact is being made. So this early point of contact seems to be also a spot where these receptor subunits seem to be expressed the earliest. Beta 2 and beta 3, like I mentioned, beta 3 seems to be expressed earlier than beta 2, as you can see here. And eventually, both of these subunits are expressed pretty widely throughout cortex. There's also some layer specificity. As you can see, the deeper layers seem to be expressing less of beta 3 in particular than the superficial layers. That's not quite the story for beta 2. And a lot of this laminar specification that we're finding in these studies will definitely take us more time to understand the functional significance of it. And laminar, by the way, in case I'm using a complex term, that just basically means different layers of cortex. And then last but not least, there's this combination of delta and alpha-4 subunits. So delta is the subunit that typically forms receptors that mediate tonic inhibition, that are outside of the synapse, mediate these slow signals. And so what we see is this subunit is expressed quite late during development. So it's not really present during early development. And typically with delta, the subunit that partners with it to form receptors is the alpha-4 subunit. And you can see that the expression of these two is very similar. They both are pretty much gone in developing brain, and then they start being expressed later. So most likely these also are not as important for the developing brain, and a lot more important for the function of the brain once it's mature. Okay, so these are the two main methods by which the brain can accommodate developmental signals mediated by GABA during early development, and then transition to mature neurotransmission, where neurons are able to turn off other neurons and regulate a circuit firing in the adult brain. So to summarize it, in a way I suppose I just did, but GABAergic signaling through the GABA-A receptors plays an important role in brain development. I hope that's a big takeaway message you can take from my presentation. And these signals change as the development progresses to accommodate that. Basically, GABA is excitatory during early development, but it later becomes inhibitory in later development and with maturity of the brain. And one way that occurs is by this transporter KCC2 becoming active. And then GABA-A receptor composition also changes as development progresses. So early during development, there seem to be a lot of alpha-3 and alpha-5 containing GABA-A receptors. And then later in development, there seem to be a lot more alpha-1 and alpha-2 containing receptors, and then also the alpha-4 and delta containing receptors. But so much is being studied, including, the significance of these processes specifically to development or to specific developmental processes. So a lot still needs to be learned. And with that, I'm going to close. Thank you very much for being here and listening to me. If you have any questions, I'll be happy to take them. And here's just a kind of simple schematic of what GABAergic signaling might look like, including both synaptic signals. So again, those are going to be mainly alpha-1, alpha-2, alpha-3 with gamma, like I mentioned, and the extrasynaptic, in case that was confusing when I was talking to you earlier. Here you can see it on the schematic. You can see certain combinations going to be outside of the synapse. They're going to be mediating these slow currents as opposed to these quick currents. And last but not least, you see in red, we have the GABA-B receptors here. I didn't really talk about them at all today, but this is another way that GABAergic signaling can occur. That's just not something I studied, but it is something that can be quite important and just something to keep in mind when we talk about GABA. All right, thank you.
Thank you so much, Kirill. That was such an amazing presentation, and we have 7 questions waiting for you. And like you mentioned, a lot of information, and maybe some of these questions may need some more exploration. But so here's a question. When considering someone who has a GABA-A mutation, such as alpha-3 and alpha-5, when subunits are expressed early and then re-expressed through a treatment, is there a theory that the development processes could be rescued later? And then the second part of that question is, you mentioned that the development of the layers can be reintroduced. Hold on, let me reread this. And you mentioned that they develop in layers. Can you reintroduce the expression in those deeper levels?
So development is a very unique time. During this time, you have a lot of different developmental processes going, and differences in expression, and it's very hard to recreate that later on. Specifically, one good context in which I know it is stem cell therapy. For instance, for epilepsy, there's right now actually a clinical trial where they inject GABAergic interneuron progenitor cells into the hippocampus of patients, and they've done it. There are actually some really cool stories. Like there was like a sea lion who had epilepsy, and they didn't have this drug yet approved, so they did like an experiment where they treated the sea lion's epilepsy, and it actually went really well. The sea lion doesn't have epilepsy anymore, and they're in clinical trials now. But so there, a big question is, if you take these neurons that express, in the context of receptors, let's say, they will express GABA-A receptors that are probably appropriate for development. And in context of KCC2, they probably don't express it yet, so the signals that they will receive will be excitatory. What ends up happening? Can they actually navigate a mature environment and actually integrate into circuits there and act as if they would during development? I think the experiments that have been done to that effect show that this happens in a somewhat limited fashion, but of course, obviously, it does work in that context because those cells are able to integrate, and they basically increase local inhibition, and they're able to rescue excessive firing in that circuit. But in other cases, it can also be very risky. For instance, in case of traumatic brain injury, what's been shown is after traumatic brain injury, particularly hippocampus in studies, that whole area where injury occurred, after injury, it goes into a period where KCC2 is not expressed very much, so GABA becomes excitatory once again. And the receptor composition that's expressed goes kind of similar to what is expressed during development. And you have increased birth of cells in hippocampus, increased birth of neurons, because hippocampus is a unique place where adult neurons are still being born in the adult brain, which is the very few places where that occurs. But in that context, what actually happens is people after traumatic brain injury and animal models, they can develop epilepsy. And actually preventing birth of new cells in the hippocampus can reduce the risk of them developing epilepsy. And that is because those cells don't integrate properly into the circuits. They form this kind of closed circuit kind of, it's been shown, they form this kind of a closed loop, closed circuit arrangement that ends up causing seizures later. And so in animal studies, at least, where that birth of neurons is prevented, it seems to actually help prevent seizures from occurring in mice. So, in that example, I think that's maybe the cells can get those signals, but then what ends up happening is not what happens during development, because it's no longer a developing brain, and it can actually be dangerous. In context, so those are the two examples I know that are probably the best to bring up in this case. But I think what you were asking me specifically, for instance, the deeper layers are formed first. Can you express certain subunits in those deeper layers? I mean, mainly what I was talking about is newly formed neurons. So whether you could go to expressing something else in a neuron that's already mature, convert it to a progenitor, that's even less likely. If a neuron is there, send out its processes, connected to other neurons, you're not going to be connecting it. I mean, you're not going to be making it an immature neuron anymore. Could you possibly convert it to a young neuron state, to where it will make more contacts with other neurons? It'll start sending out neurites again, maybe connect better. I'm not sure about that. I mean, you can possibly somehow increase plasticity, but I'm not sure. And I'm not sure regarding the, reverting back to expressing specific receptor subunits. Did I answer all of that, or was there some other part to that question?
No, that was great. And I definitely want to talk about that with you a little bit further at a later date. But I have 3 questions that I'm going to combine together because they really just complement each other. So it goes like this. How might modulation of KCC2 expression or function be leveraged as a therapeutic approach for individuals with GABA-A mutations? Are there any existing drugs or compounds that target KCC2 or related pathways that could be repurposed for treating GABA-A mutations? And what preclinical or clinical studies are needed to further validate the efficacy and safety of targeting KCC2 for treating GABA-A mutations?
Okay, so that whole even going slightly wider than KCC2, just the aspect of GABA being excitatory during development. It's been a pretty hot topic for over a decade, I'd say. One of the things that has been done is, so there is a drug called bumetanide. I think it's used for originally, I want to say urea cycle disorders, but I don't remember exactly. Or I think it affects kidney function somehow. But basically, it doesn't target KCC2, but it does target NKCC1, which is that transporter that moves chloride into neurons. So if you give that drug, even before KCC2 is expressed, you can have an effect somewhat similar to what you get with KCC2 expression, because you prevent those neurons from getting loaded with chloride. And so GABA isn't quite as excitatory. And in one of the experiments, or several even, I think that I showed here, that's one of the drugs they used. And they saw that it had an effect on developmental processes normally promoted by GABA. So there has been actually a clinical trial with bumetanide. From my understanding, and I think it was to help with like early seizures, where they thought maybe these kids, they had early seizures because GABA wasn't actually inhibiting things, but was instead only helping the seizures by being excitatory. And in that context, they gave bumetanide, and those studies didn't give very good results. But I don't think research groups, at least certain research groups, entirely gave up on that. I think that's still being investigated, but there have been some clinical trials with bumetanide. Then KCC2 also has some attention. And by the way, so we're obviously talking about GABA variants here, but also there are mutations in KCC2, which are also associated with neurological disorders, including epilepsy in people. Right now, there are drugs that are being developed actively to regulate KCC2 activity. I think both to activate it and deactivate it, different drugs. They're actively developed right now. I don't think any of them are actually used for clinical trials right now. I know one of the biggest groups working that is Stephen Moss's group. And then there's another one here at Vanderbilt. I actually collaborated with them a little bit by David Weaver here. But yeah, but to comment on where they come, how far they come in the clinical context, and what the exact hypothesis there would be, especially outside of a disorder that actually involves KCC2, for something like a GABA-A receptor disorder. That's really sit and think through it, because from what I understand, the polarizing GABAergic, excitatory GABAergic input is very important during development. So you probably wouldn't want to get rid of that. But then inhibitory input is very important in the adult brain. So you also wouldn't really want to tamper that. You might be able to kind of modulate just how inhibitory or just how excitatory things are by modulating how much drug you give and modulating the effect of KCC2. But I mean, the effect of like, if you turn off KCC2 just a little bit, could you make all signals be just a little bit less inhibitory if you have too much inhibition in case of like a gain of function mutation? I mean, maybe, but that'd be such an isolated effect. I think it could really wreak havoc on the brain in terms of the systemic effect it'll have on doing that to all the neurons. So it's kind of hard for me to hypothesize there. But the tools are getting there. There are people working on it. And if I were to sit down and start reading up on it more, maybe I would find a more specific answer to your question.
Well, I know that you are working on it. So is everyone at Katty Kang's Lab, and we're so grateful for the work that you're doing in lab.
I'm grateful for you having me. Yes, Martina?
As you mentioned, sorry, interrupting. As you mentioned, bumetanide. That is interesting because that's a medication that we tried long time ago. It was 2018, and it was tried because my son had such an opposite reaction to clonazepam. And as we didn't see, some major things on bumetanide, after 6 months we stopped it. But it was really, really stable period. Like, he was walking better, like looking healthier. So I don't know. Now, going back and hearing about it, it was maybe something that we should give more time to it. So maybe the researchers will test it on our variants. Something to reconsider. The complication is that we have G6PD deficiency, and this is a sulfa drug.
Okay.
So,
and by the way, as your son gets older, I assume the effect of bumetanide is going to be less and less, because the brain will be basically expressing more and more KCC2, and GABA will be more and more inhibitory.
Oh gosh, this is so complex.
Unfortunately. Yeah.
Thank you.
I understand also that, to me, this is something I study. So when the brain is very complicated, I can kind of laugh at it and keep building models and researching it one bit at a time. But you're very difficult situation, fortunately, where you're trying to find out how to help your son and how to navigate in this very difficult environment. So I'm sorry for what you have to go through. I really wish I could give you more specific answers.
Well, we had already, I'm very thankful for all the researchers that doing, because the gain of function, knowing about gain of function is huge. It's really huge. Even avoiding specific medications and navigating this from this side is critical. So
Mhm.
super thankful for all the efforts and work. You're making a difference in many families' life.
Well, I
Thank you.
appreciate you coming out and talking, and so I can hear your feedback, and just being a very active parent. I don't know if everyone does that, but and just basically the whole story you've been telling me, I can see how proactive you've been, and going to doctors and just learning this and researching this and thinking about it critically. I think that's great. I think hopefully that will, come through and help your family in the end.
Thank you. Thank you, Monica.
Thank you so much. And if anyone has any more questions, please send me an email and we'll send them all off to Kirill at the same time. Thank you so much, Dr. Zavalin. We really do appreciate it.
All right, great. Thanks, everyone. Have a great day.
Bye.